followed by again 20 to 30 hours of division, and then it will be 3 cell stage followed by 4 cell stage. Now why 3 cell stage, and I have written there transient? Reason is it has been found that the zygote when divides, it divides into 2 unequal sized cells. So one cell is slightly bigger than the other, so obviously the smaller cell is going to finish its next division faster compared to the bigger one. That's why there will be a temporary 3 cell situation which will be definitely changing into 4 cell situation. Now all these divisions collectively are called as cleavage. Now you need to understand the difference between a normal mitotic division and a cleavage. What is a normal cell division? A division in which, if you go back to your 11th class topic of cell cycle, teachers have taught you already there, G1S and G2 topic. So what exactly happened to a cell after its formation? It should enter into G1, then only S and G2. But now what exactly happened in the cleavage? There is a very small size of interphase. If you remember, interphase is 95% of the cell cycle time is occupied by interphase. Here it is going to be a very short duration. Reason, the cell is not going to grow bigger in size. That means, but the zygote, the size of the zygote will be the bigger one here. It will divide into 2 smaller cells. Please remember the sizes, the volume of these cells is decreasing, but the surface area is going to remain same. The reason is that because the cell is trying to bring its nucleocytoplasmic, or you can say the kern plasma ratio to come into the right proportion, because if the volume is greater compared to the surface area, then the cell will find difficulty in controlling the rate of metabolism. To have the best rate of metabolism, a cell should always have a right proportion or a right ratio of the surface to the volume, or the ratio of the nucleus as well as the cytoplasmic ratio. So these ratios have to be the best suiting ratios so that the cell find it comfortable to perform all the metabolic reactions efficiently. So this zygote is quite a big cell. Reason, if you remember, sperm is very small, 60 micrometer was the size I told you, and the egg is going to be around 100 micrometer. So this cell is quite big compared to a normal human body cell. So its size has to be decreased to such a level that it becomes the right type of size to perform all the metabolic activities efficiently. So the nutshell is, but during cleavage there is a very small duration of interphase, or you can say that there is a minimum growth of cell, or growth is absent. Only the cell is going to prepare things which are required to enter into the next division. So there is hardly any change in the volume of the cell. So cells are becoming smaller every time there is a cleavage. So now these 4 cell situation is going to divide further into 8 cell situation, then 16 cell stage, and these stages are called as morula. Morula word comes from German language. Morula, morus, if you remember, is a scientific name for the mulberry or shahtut. So this looks like a cluster of cells which look like shahtut type of structure. That's why it is named as morula. Sometimes morula may even carry 32 cell situation. And finally, you can see in this picture there is a zona pellucida still surrounding. I have not drawn here, but zona pellucida still surrounding the morula, and there are 8 to 16 or maximum 32 cells present. This happens by the end of the 4th day. So please remember, this happens up to the end of the 4th day after the meeting, or you can say after ovulation. Now this 32 or 8 to 16 cell stage is going to change into the next stage. And what is the next stage now? Blastocyst. Now please remember, all these cell situations are called as blastomeres. So zygote will first change into 2 cell situation of blastomeres, then 3, then 4, and finally morula, and then blastocyst. Now what is blastomere? Blast means to form, and meres means the monomer. So it is a fact that if you take any of these 4 cells